welcome to this session. This special I3 session today is going to be about the quality inside, about how technology is helping quality management in the IVF lab. This session is sponsored today by the Reflections app from IVF QC. And when we think about quality management, what do we think about? The ability to create an optimal environment for embryos in pursuit of successful outcomes for our patients is largely based on quality management. Quality management, as we all know, encompasses quality control, quality assurance, and quality improvement. Most of us perform these tasks every day towards this QN program for our labs and clinics. As embryologists, we perform daily QC of temps, liquid nitrogen levels, incubator gas checks, warmer temps, CASA calibration checks, etc., all to ensure tight quality control. Lab directors and supervisors look at tech competency, compliance with SOPs, document control measures, PT events, we all have to do that, outcome measures throughout selected KPIs that we choose to look at, staffing levels, which I'm sure all of you guys would like to talk about today, and patient experience surveys, all to ensure that the quality of the program remains optimal. Today, we are going to hear about how technology can help us with that quality management. All of the contact information for the speakers today and the extensive long bios can be found on the site at www.ivfmeeting.com. We encourage you to please ask your questions again in that Q&A section that's found at the bottom of your screen under Q&A instead of the chat function. Today, I'm joined by my co-host, Cassie Miller, Ovation Fertility Director of Risk Management. She helps Ovation's IVF labs maintain regulatory compliance, and her primary focus is ensuring that Ovation's employees have what they need to work safely, the embryos are protected, and that patient identification and privacy are maintained.
Thank you, Jean. Jean Popwell, my co-host here today, has worked extensively as an embryologist, a trainer, a lecturer, a consultant for over 20 years. As a lab director, QA, QC director, Jean is responsible for the management of the quality assurance program for the Prelude network of ART labs. Jean works to research best practices, document SOPs, and collaborate with the lab directors of the lab steering committee to implement quality standards. Our first speaker today needs no introduction to this crowd who know her as a distinguished scientist, devoting 35 years as a clinical embryologist, focusing on improving IVF laboratory performance and outcomes via meticulous practice, detailed observation, embracing of new and bold technologies, and teaching and training of others to do the same. But now also as president of the Alpha Society of Reproductive Biologists. Welcome, Mina Alikani.
Hi, thank you very much for that introduction, Cassie, and thank you to the organizers for inviting me to make this brief presentation on quality control in the IVF lab. I do want to apologize. I'm just getting over a terrible flu, and so my voice may be a bit rough at times. So, like most practitioners, for years, my laboratory's quality control routine included collecting the data on paper, filing these reams of paper in binders, and storing these many binders wherever we could find, and around the offices of the laboratories. And then when the time came, presenting the binders to an inspector, and following that, discarding all the paper after the required 2 years of maintenance on site. I did review and sign those reams of paper as the lab director, but there was no straightforward way to analyze the data or keep historic tracks, certainly not without major duplication of work via entry onto an Excel sheet, for example. But there were many issues with that system, one of which is well demonstrated on this QC sheet. At first look, you would say, well, warmer 1, which is the instrument being monitored on this particular sheet, seems to function quite consistently, showing the same exact temperature of 38.5 degrees, though it's unclear whether these are display readings or measured readings over the entire month of November. All the readings were initialed and the supervisor signed off on it. And so is everything in order? Well, the answer is no, not quite. If you pay close attention, you will notice that the expected range for temperature in the case of this particular piece of equipment is indicated as 36 to 37.5 degrees C, which would make all of these measurements out of range. And as you can see, there are no comments or corrective actions taken in response to these out of range values. To make matters worse, and this is the same instrument, warmer 1, someone well-meaning but clearly uninformed in terms of what QC actually means, then changed the ranges to read first 35 to 40 degrees and then 34 to 40 degrees, both of which are quite unlikely to be the correct ranges. I can't see the audience, unfortunately, but I'm sure some of you are cringing at the moment, and some of you are dismissing this as other people's problems and not yours. And as some of you may think, oh, I've seen that before. The fact is that this is neither unique nor infrequent. If you use a clipboard and printed copies of your mostly uncontrolled forms, this happens more frequently than you think. So the solution, I would argue, is not to find better clipboards, as this ad is trying to convey. It is to ditch the clipboard, because the clipboard belongs in the same category as all these other objects, obsolete in 2022. So now let's talk about quality control, why we do it. Is it A, to make ourselves feel better? B, to pass inspections? C, to please the lab director? Or D, to ensure safety and quality of the care we provide to our patients? Well, if you answered D, you would be correct. Although if you answered all of the above, you might still get some credit. We do quality control in order to ensure the safety of the procedures and the services that we provide to our patients. And of course, gametes and embryos in the laboratory are our patients. If you Google the meaning of quality control, and in particular looking for the difference between quality control and quality assurance, you will end up a very confused person. But in a very simplistic way, you can think of quality control as being a component of quality assurance, which is itself part of the quality management system. One way to think about it is quality control is the collection of the data, whereas quality assurance is the analysis and interpretation of the data, and using the data to ensure the high quality of the products that you are producing. IVF-QC's Reflections app is a cloud-based tool for quality control and quality assurance. Data are collected directly into the app using an iPhone or an iPad or a computer, whatever is your favorite device, and stored in the cloud for an unlimited amount of time. The files can be backed up digitally, locally, and kept indefinitely as well. There's no need for the use of paper. There's no need to print anything and sign anything in person. It can all be done digitally, and its intuitive design makes it very easy to use. Why I say it's a tool for QA is its ability to generate detailed reports, including fluctuation graphs for any parameter for any period of time. It also has the ability to track media and consumable batches. I introduced Reflections into one of the first labs first in one of the labs that I was overseeing back in 2015. And I have to say that since then, I have implemented it in many more laboratories with outstanding success. So Reflections does all of this. So lab directors like me and lab managers could sit on the beach with their minds at ease that everything is going well in the lab. Of course, I wished. But the way to use it is rather straightforward. You define an activity and the frequency at which you would like to run that activity. You specify the equipment, parameters, checklists, etc. You establish ranges for each parameter. Data are entered real time or streamed from your equipment or devices. And then you can get summary reports for any desired period, as I mentioned, and get fluctuation reports for any desired period or any instrument. You can note corrective action measures right there in the app when you find out of range values and they need some kind of response or corrective action. And you can even file incident reports. It will all be in one place, and it can all be reviewed and signed off electronically. So Reflections is organized according to three broad categories: instrument type, instrument, and parameters. So in this system, an example of an instrument type is incubator. And examples of instruments will be different types of incubators you may have on hand. And for this particular instrument, which is the incubators, the parameters you measure or record would be gas levels and temperature, possibly gas pressure, and the values that are measured or displayed. And you can specifically indicate which of the two you are entering. So it's very clear. Was it the instrument value that you are reporting, or is it something that you are actually measuring using a device of your own? So in the embryology lab, for example, of course, this could apply to any kind of laboratory, any kind of facility that requires quality control. And so in the instrument types in embryology happen to be things like incubators, refrigerators, freezers, cryostorage dewars and tanks, the environment, temperature and humidity, tube warmers, stage warmers, biological safety cabinets, workstations, And then you can also create checklists as instruments. So for example, if you are looking at tank, cryo storage tank integrity, you can have a checklist and you can include that as an instrument. You can also have things like a shutdown checklist, which again, it's not an instrument per se, but in this system, it is treated as an instrument and set up exactly the way you would like to see it. So the parameters in an IVF lab are rather limited, as are the devices we use to make these assessments and measurements. You may use a thermocouple, a gas analyzer, a blood gas analyzer, an osmometer, and dipping sticks to make measurements, to make measurements of ambient temperature, ambient humidity, VOC levels, CO2 levels in incubators, O2 levels in incubators, temperature, pH, osmolality, door openings, the number of door openings in the incubators, and gas cylinder pressure, for example, in your manifold room, liquid nitrogen levels, and all the other checklist items. What is nice is that alerts are created every time an out of range value is entered into the app. And you can set it up so that you immediately receive a text message as a lab manager or a lab director, or even others in the lab, including supervisors, can set it up so that they can immediately see that an out of range value was entered. And of course, this is not to say you need to panic. It just means that something was out of range and some action may be indicated. So when you go into the app, the dashboard shows you the logs, which are really the plans of quality control activity selected for each day of operation, and whether or not they have been completed. And with one glance, you can actually see whether the intended QC plan for that day has been completed. Under reports, you will see the summary reports, which can be generated as often as you like, but for regulatory purposes, once a month at a minimum, and signed by the lab director or supervisor or manager electronically. And you can obviously save all of these files or delete them if you don't want them, etc. There is quite a bit of control. Under fluctuation and detail, you can actually graph over whatever period of time you choose, any parameter that you choose. So if you want to look at, let's say, room temperature between October 3 and October 27, you can actually plot that and see whether there are any trends or potential problems. You will also see a section on integration, which, because of an open API, allows integration of other systems and streaming of data. Now, this is an example of a piece of equipment that actually keeps its internal data continuously. It's an embryoscope that keeps track of oxygen levels, CO2 levels, temperatures, pressures, and all of the data are available through the server of the embryoscope. But if you wish, you can actually have this streamed into reflections. And as I hear from Marianne, there's now a possibility to autofill specific time point values in reflections using the streamed data, because of course it's an enormous amount of data, and how to manage that on its own is a challenge. But if it is possible to select specific time points and then have those filled in automatically in reflections, of course, this is an enormous help. The same thing with a system like the K system, where there's a software called K-Link that keeps track of everything. Again, CO2 levels, O2 levels, any alarms, pressure, nitrogen pressure, CO2 pressure, etc. All of that is kept track of, and how wonderful it would be to be able to use that data and stream it into reflections so that you have everything in one place. So have we resolved all of our problems then by introducing an app like Reflections into the laboratory? And the answer is no. We still have some unresolved issues. Integration is an issue. It's an important and big issue. We have too many independently operating systems that are not necessarily interfacing and interacting. And so we need to find solutions, as IVFQC is, to that issue. And then there's the question of how should continuous monitoring modify periodic monitoring? Honestly, I have not really found the answer to that question, mainly because of something that I have also pointed to here, which is that the regulatory bodies are still a bit behind times. And at least in the US, they are still unfamiliar with some of these newer concepts and are expecting to see the same thing that we did for the huge box pharma incubators of the 1990s. They expect to see us do the same thing that we did back then with the highly sophisticated embryoscope or the K system or the MIRI or the Aztec or many of these other newer versions and generations of equipment that are far more capable of keeping track of the key parameters of their functions. So how much data is too much? We can also end up having too much data. Where do we draw the line? And how do we keep quality control comprehensive but not draining of our limited laboratory resources? And then ultimately, it's the number of QC activities related to outcomes. And I think that Giles Palmer and colleagues did a very beautiful study a few years back where they looked at some aspects of the number of parameters being used and the data points being collected by different laboratories that were using reflections, and came up with some very interesting observations. I'm not going to get into that, but that question or those questions are still relevant, and we need to be able to answer them. Thank you so much for your attention, and I'm happy to answer questions.
Thank you for that great presentation. Dr. Alikani. I do have a question from Minji from Zurich, Switzerland. The question is, what education background should one have for a person responsible for the QM in an IVF lab? And what is the daily average workload for that person?
Did you say QM, quality management?
Yes.
Yeah. Okay. So I think that someone who takes on the role of a quality management officer in a laboratory should be familiar with all aspects of quality management and has to understand a quality management plan, and perhaps even in collaboration with the laboratory director or manager, be the one to write this quality management plan. It's extremely important that you have a very specific plan that addresses all aspects of quality management. And that includes document control, proficiency testing, continuing education, quality control, quality assurance. So it's a very broad area that requires a lot of knowledge, hands-on knowledge, practical knowledge, as well as theoretical knowledge on how quality management works. And then, you use that knowledge to customize a plan for your own setting and your own laboratory.
Thank you. And we'll have some more Q&A later for all of the speakers. But now it's my pleasure to introduce Rusty Poole. Dr. Poole is the Scientific Director for the Fertility Center of San Antonio in Texas. Dr. Poole has held this position for 37 years. Prior to this, he was an associate professor of cellular and structural biology at the University of Texas Medical School in San Antonio, where he worked on culture and cryobiology of non-human primate embryos. He is the 2018 recipient of the Lifetime Achievement Award from the SRBT of ASRM. And if you want to know more about his extensive, fantastic career, please do check out that long bio at the site ivfmeeting.com. Thanks, Dr. Poole.
Jean, thank you very much. That was a very kind introduction, and thank you and Cassie for your excellent moderating skills, and thank all of the people at I3 for inviting me. It's always a pleasure to be here and participate in these webinars. I'm going to talk a little bit about quality control and use of cloud-based systems that actually can bring quite a bit to the table in terms of converting it to quality improvement. I have just a few disclosures. None of these will affect anything I talk about. Well, as you just heard, Mina did a fantastic job of talking about the Reflections app. Not surprising, but I want to tell you, Mina's also done another fantastic job, and that is about 8 years ago, she senior authored with her colleagues, Kathy Go, Carolyn McCaffrey, and Dave McCullough, a fantastic paper that if you don't have this one in your library, you should. What they attempted to do is to look at the contemporary art laboratory setting and try to evaluate it in terms of staffing guidelines, and followed it sort of as the IVF lab chores have evolved over time. It's a fantastic paper, but one of the things they did, I think in a really comprehensive way, is to look at what we do in the lab. Now, this next slide I'm going to show you, please don't try to read it. This is just look at blocks. Blocks here represent, in this diagram from their paper, procedures and adjunct procedures performed on a daily basis in the contemporary IVF lab. I'd like to point out, I think there are 56 blocks here last time I counted, but this was 8 years ago, so it's grown. So there is absolutely no doubt embryologists don't even have a chance to breathe during the day because of this. So time constraints is of the ultimate issue to working effectively in the IVF lab. And we don't have time to do a lot of extras. So we have to be exceedingly efficient if we want to do that. A second paper I'd like to point your attention to, and Mina just mentioned it. This was published about 3 years ago in RBM Online. The senior author was none other than I3's Giles Palmer, as Mina alluded to, and it contains as co-authors some of the I3 luminaries, Marianne to be one, her brilliant daughter Sarah to be another, Jacques Cohen to be another. And as Mina said, they looked at the Reflections Act as it was applied to 36 different ART laboratories that spanned over 8 countries. Now, these were laboratories that had at least been using the app for 6 months. And so they sort of were familiar with it and knew what they were doing. And as Mina said, a lot of really good data came out of this paper. Very interesting observations. I'd just like to point to a couple of them, though, for our purposes. This is a distribution of those laboratories, and the bars represent the number of parameters that they recorded and reported to the Reflections app. 2 things pop out, at least to me. First of all, most of the laboratories collect very, very few parameters. And of those that collect a bunch of them, those are typically, as Giles and his colleagues pointed out, from areas where certification, licensure, inspection is required. So it tells us that QC largely, at least as of 3 years ago, was something we did because we had to, not because we had time to or wanted to, or maybe even didn't see much value in doing it. And but with this app, I think the time has changed. So QC is now something that we can deal with in a very logical way. And I think putting it against that background that Mina and her colleagues pointed out, do it in a very time-effective way as well. So what I want to do today is to talk about quality control and use examples from other industries besides ours that have utilized cloud-based QC and their ability to convert that into very usable improvement data for whatever business they're in. Mina did this fantastic job of talking about the pitfalls of clipboards, the binders, local servers, et cetera. Cloud storage, there's a couple other quick advantages, though, that I'd like to talk about. One of them is it's very economically efficient, and that is to say, you only pay for the space that you use. You buy a local server, you have to buy all of that storage capacity up front, and you don't use it until the server's full. Cloud storage, you pay for only what you use, and it's expandable. You can get it increased daily or shrunken daily, and again, you're just paying for what you use. The other thing is just a quick mention about cybersecurity. I mean, the cloud is not innately any more secure than any place else, except that the cloud purveyors, the people that will put you onto the cloud, typically offer you as a client a robust toolbox of cybersecurity measures that give you a much better level of assurance than you would get locally. It's not perfect, but it certainly is a much better deal than you typically can get locally. So it's secure. I do just to quickly point out, if you are breached, if you've been hacked, it typically takes an average of 200 days to know you've been hacked, and then another 70 days to deal with that. And if you're ransomed, or if your data are ransomed, and you make the decision to pay the ransom, you've got an 80% chance you're not going to see your data anyway. Remember, these people were crooks on day one, and they're crooks the day you give them the money for the ransom. So it's best to really think about a very secure environment. And finally, you may say, well, look, what are they going to do with QC data? Believe me, QC data comes in the level of commercial espionage, and they would use the data. So it's a very good idea to have your data secured, as little as you may think QC data might be. So who uses a cloud-based QC? Well, any mature industry you can think of. Here's a laundry list of them, from food and beverage industries, manufacturing, automotive, aerospace, medical, pharmaceutical, name it. They do it, and they don't do it just as a place to archive what they've been doing. I mean, I think Mina did a great job of showing that we've looked at QC many ways as just being an archive of the things, the way that things were at the time we collected the data. These industries, they do archive their QC data, but they don't do it just for that. They do it for improvement. So I'm going to give you just a quick look at a couple of examples of the ways people are using it. Here's an interesting one that I found. This is a company that offers a QC on the cloud to companies that use media. These are filmers, video people that you can use, from filmmaking to advertising. Any way you use media, you can take your quality control data and through this company upload it onto the cloud. It's pretty cost effective to do that, but because you can tell them how to analyze your data, you can set templates, you can get automated objective testing of your data. I mean, it also serves as a repository, but then upon inspection, it can find errors, it can correct the media. It can enable agreements that you may have and give you an audit trail. So it gives back to you a much cleaner picture of the data that you're producing. The second one, which is I found really interesting, this is a manufacturing example. This is a company that manufactures roofing materials. Now, if you are a manufacturer of roofing materials and you, on your assembly lines, your product lines, you want to inspect for quality, and you're going to do it manually, it takes a tremendous amount of manpower. You're going to look for line spots, holes, color problems, anything that discrepant in the product. It's going to take a lot of man hours and money to do that. There are, however, sophisticated optical methods using machine vision that can do these kinds of analyses and do them in a very effective way. The problem is, if you want to integrate that on your manufacturing site, it costs a goat farm to do that. It really is not very cost effective to do it. So here's a company called Intrinsic Imaging. It's a California-based company that offers these analytics in a cloud-based system. So all you need as a manufacturer is to have a camera that will communicate using an internet protocol. So an IP camera is all you need. That's very cost effective. You can put it into your fabrication line, and you can get real-time feedback on your data that can analyze your product for just about anything you want to. And here's an example of that particular system. So roofing materials can be produced on a production line. And in that production line, you can capture images, again, on an IP protocol camera, run it up in a secure manner to the cloud. Now, this particular company uses AWS, which is the Amazon Web Services system, as their cloud purveyor. And in there, resident with this company is all of the expensive analytical equipment that you can use to analyze your data. So you get several different places wherever you want pass-fail answers to different quality problems you might have. You get that quickly, and it's fed back almost in an instantaneous loop back to your manufacturing line, so that products that don't meet your quality standard are eliminated, and your buyer never sees it. So this is instantaneous quality improvement by jettisoning the labor that you would do by manual and putting it up on the cloud, where you can now afford this analytical equipment. It's a very comprehensive and sophisticated, but expensive. It's also been used in systems involving laboratory analytics. Excuse me. If you are having a proteomics laboratory and you use mass spec, here is a system that was published several years ago, where you can upload your data directly from mass spec into the cloud. There are analytical capabilities in this company that will then do all of the QC metrics that's required for this particular data, so that you can put quality in your analytics of proteomics. And so it's very cost effective. I think really, if you just use a little bit of vision as embryologists, I think we can see that we could integrate systems like the manufacturing example I just shown, and like this one, into our laboratory. I mean, we're basically doing it already for novel ways of doing embryo selection, but you could integrate it into producing quality systems. For example, I could envision putting it into the system where we set up our culture platforms with the appropriate kind of analytical equipment, and using cloud-based sophisticated analytical instrumentation. We could analyze for medium quality, oil quality, dish quality, drop volume, oil volume, et cetera, and produce an exceedingly routine and reproducible culture system that manually is very difficult to do. That's just an example. So I think there's lots of ways we can go to industry and pull out these ideas and apply them in our setting to produce good outcome. I'm going to come back to the system that Mina described. We've been also using the Reflections app for probably about 6 years now. And there's some features of the app that I really think are very nice. Here's a quick example. Actually, this was offered in an earlier session that was chaired by Giles on the Awesome IVF lab meeting. We had in my lab several years ago a scenario where we kind of fell off in the production of blasts from the 8-cell stage. So that's something that we monitor, among other things. And it had dipped down a little bit. But one of the beautiful things about the Reflections app, as Mina was pointing out, you can just with a few keystrokes pull out any parameter and any data point you want to look at for any instrument for any period of time with just a couple of keystrokes. It would be impossible to go in and try to interrogate your binders to get this kind of data. but with just hitting the return button on the computer, all of a sudden you get the data displayed in a shoe heart chart, and you get statistical treatment of the data as well. But what I noticed in here, and this is the period of time, this is back in 2020 that we were experiencing issues. This coincident with that, we were seeing, now this is a digital readout on the incubator, a little rise here, a little wobble in temperature that we don't see the rest of the time. This is a real very tight temperature range here, but you can see that this is different from the rest. So if I went down and then looked at the measured temperature that we did, you could see there's quite a bit of wobble in this area. And trying to decipher this, all of a sudden it hit me. You're looking down at the time frame. This is late May, early June, and it persists into late August and even into getting to September. But ultimately it stabilized when we got later into September, moving into October. Well, I'm going to tell you, this is South Texas, and it's hotter than blazes during this period of time down here. Our building, the professional building that we're in, likes to chill us to death. Our employees have to wear a jacket during this period of time in our center. So temperature regulation in the laboratory is much more difficult. But this pointed out to me that this isn't just a subtlety. Notice nothing was out of range, but you can inspect data here from different parameters, and I think get a lot of insight into what might be happening in terms of performance in the laboratory. So it's just one simple example. And with that, I'll just stop. I'm going to end quickly here by relaying to you a brief anecdote. I think it was about 20 years ago, Jacques Cohen produced a series of 3 embryology meetings, really some of the best meetings I've been to. And he spaced them 2 years apart. He was doing a biennial review of embryology. The first 2 meetings were held in Mexico, and I was invited, I can't remember if it was the first or second meeting, to talk about cryopreservation, which I was happy to do. Well, in those days, most of the time I was talking about equilibrium cooling, slow cooling. But at the end of the talk, I got into vitrification and very excitedly said, look, you don't need to have expensive equipment. It doesn't take you a couple hours on a per embryo basis. It's cheap. It's very, very quick. You know, that it's all better for the embryologist, and closed out. The very next speaker in that meeting was one of the brightest embryologists that's ever walked the face of this earth. That's Steen Willitson. He came to the podium and he was irritated, and in a very vociferous way began to tell the audience immediately, it doesn't matter if it's faster, cheaper, easier. None of that matters. The only question is it better? Now, if I had had a Harry Potter invisibility cloak, I'd have used it. But I have to say it was a great lesson, because he's absolutely right. It doesn't matter if we integrate new technology just for technology's sake. If we use more sophisticated technology, we need to know first and foremost, is it better for our patients? That's the end of the game. And I think we're getting those tools now to convert QC data into quality improvement data in a very time-effective way for the stressed embryologist. And so with that, I'm going to stop and say thank you very much.
Thank you, Dr. Pohl. That was very informative. I always enjoy speaking with you. I think you know our history. Introduction was I was in sales. I would sit in your office and we would just chat for hours about science and innovation. And so listening to you today, I listen to, you know, what my staff says when they're looking at QC. We've just moved into a reflections platform, to me, which is such a huge move from just taking data, putting it on a piece of paper, and then moving that into a binder where you can actually see, you know, it graphed out. You can see shifts and trends. You can go back and troubleshoot. You know, for me, as working with researchers, and we love data and we love KPI and we want better blast results, you know, teaching or training our staff to look for these shifts and trends to see, was the incubator acting differently because we had, you know, the air conditioner turned up during these months. You know, listening to you talk about the technology, and we have all of this artificial intelligence, perhaps what we as humans can't see when looking at this data, maybe we need to move, and I wonder if that's what you're thinking too, into some kind of AI platform that looks at our data for us and says, you might have missed this shift or trend that may be affecting your KPIs. What are your thoughts on using AI to review our QC with all this data that we can collect?
Yeah, absolutely. Cassie, can you still hear me?
I can.
Okay, good. I've lost my screen here. Yeah, I know. Absolutely, that's where it's going. I think it's a great idea. And the thing is that right now, we still don't know what information to collect, what's important, what's not. We finally now, with this tool, I think, have the ability to look back and try to identify what's the most important data. You know, we really don't know. You know, looking back at the paper that Giles published, and those were pretty scant numbers of parameters that were collected. I know now in my lab, we don't do it every day, but over a week's period, we collect 78 different data points that we enter into reflections. I'm sure that's overkill. I know we probably don't need it all, but down the road, we have to have tools, as you're suggesting, to direct us into which are the best. And then we can set up through AI, whatever, the standing queries as to how we want to manage data and produce quality every day in a very measured and reproducible way. So that's absolutely where it's going, in my opinion.
Well, thank you, Dr. Poole. As always, it's such a pleasure to listen to you speak. You always have a lot to add to the conversation, so thank you. Now I am going to move on to introduce Gerardo Mendizaba. He received his PhD in computer science from the University of Houston in 2012. He is currently a research scientist at the University of Guadalajara, Mexico, and his interests include artificial intelligence, deep learning, big data, pattern recognition, and computer vision. Moreover, he is the head of product development for IVF 2.0, where he is in charge of conceptualizing and designing AI design based on assistance that can enhance the decision-making during IVF procedures. Welcome. Thank you for being here and presenting. I look forward to hearing.
Thank you very much for the kind presentation and for the invitation. It is a pleasure for me to be here. Today, I will share with you some ideas regarding the concept of the Internet of Things, the motivation for its creation, and what I believe are the opportunities for this technology to be applied at IVF clinics. So we interact with many objects every day. Each of these objects have a set of specific functions necessarily to complete our daily routines. For example, shoes help us to walk, mirrors allow us to see our reflections, and toothbrushes help us to clean our teeth. Without these objects, our lives will be much more difficult. Now, if we could consider the possibility of inanimate objects to be smart and be able to communicate with us, they might have some interesting stories to tell us. For example, the smart shoes could register when we change our walking patterns. A smart mirror could detect new skin spots, and a smart toothbrush could take and analyze images from our teeth from inside our mouths. Moreover, consider that a smart thing should be capable of taking some action with respect to the sensed data. Then the smart shoe could modify its shape to change the pressure points on the feet to correct the irregular walking patterns. The smart mirror could alert me regarding the presence of a new skin spot and give me recommendations of dermatologists near my area. And finally, the smart toothbrush could alert me if it detects a potential cavity before it gets large enough for me to be noticed. The question is then, how can we make smart the things that are around us? Well, one of the known characteristics of smart beings is the presence of a nervous system consisting of a brain, which is a central processing unit, a spinal cord that serves as main communication channel, and a complex network of nerves that interchange messages rapidly to other cells and to the central processing unit. Based on these properties, a possibility for creating a smart thing is to give the objects the capability of sensing and registering data related to themselves and the environment. Moreover, the objects should have the capability of sending messages to communicate with other objects and with a central processing unit that can generate responses accordingly. Currently, there exists a great number of electronic sensors that can act as electronic nerves. The novel technologies in developing of semiconductor allow us to count with low consumption and high resolution sensors for temperature, light, humidity, ultrasonic waves, etc. By the other hand, the latest progress in the fabrication of micro electromechanical systems allow us to count with miniature machines up to less than 1 micrometer, that can be used to sense acceleration, inertia, pressure, airflow, humidity, etc. The signals generated by these sensors need to be processed and converted into data. These require algorithms to process the values reported by the sensors and convert them into meaningful information that can be packaged into digital messages to be sent through an electronic communication channel. In the actuality, low-cost open-source microcomputers such as Arduino, Raspberry Pi, BeagleBoard, and NVIDIA Jetsons are capable of process sensor data and use existing communication protocols. This paradigm is known as edge computing. The digital message from many independent sensors can be sent to a centralized processing unit that can make sense of the data by using existing internet-capable devices and protocols, including radio frequency networks, satellite communications, or other custom-built computer networks and protocols. Now, with the arrival of the 5G technology, there's a promise to even increase the bandwidth of the internet communications in such a way that many applications that now are considered data heavy then can be considered as real-time capable. Nowadays, the centralization and processing of data from smart objects can be achieved by using computers with enhanced storage and processing capabilities in the cloud. Examples of cloud computing services providers are Amazon Web Services, Microsoft Azure, and Google Cloud, which together are considered to have more than 60% of the market share in 2022. Now, the concept of making smart things by using the technologies is referred to as the Internet of Things, due to the evident use of the Internet Protocol for communication. The term Internet of Things, or IoT, was coined by Kevin Ashton in 1999. Today, the Internet of Things has become a buzzword that is widely used in the industry. IoT refers to a network of physical devices, vehicles, home appliances, wearables, and other items embedded with electronics, software, sensors, and network connectivity that enable these objects to collect and exchange data with each other, as well as with their human users. Now, what are the opportunities for the IoT in IVF clinics? Well, embryologists use a wide range of IVF things in their work, including pipettes, petri dishes, specialized microscopes, incubators, and advanced microsurgical devices such as laser systems. While some of this equipment might come with electronic circuits and microcontrollers, it is uncommon that these can communicate with a central unit that can gather and centralize the data to generate reports. Therefore, embryologists have the challenge of keeping track of everything that happens on the lab and with the patients. Most of the common tools employed for solving this are the use of printed formats that serve as a guidance during the procedures and that must be filled by hand in many moments during the day. Also, the monitoring of the system variables is made by hand using measurement equipment, which results are registered in other formats periodically. It is really common that this data is then passed to a spreadsheet that is later processed to generate reports and compute KPIs. Many pieces of equipment and tools employed in IVF labs are candidates to be smart. In fact, there already exists some equipment that might be considered as smart, such as the smart incubators. For example, the embryoscope, the Jerry, that can be used to monitor in real time the development of the embryos using time-lapse photography at regular intervals. Also, there exists the Tomorrow storage robot that is capable of keeping a secure register of all the cells that are stored. But perhaps the first generation of IVF, Internet of IVF Things, could be considered the currently available assistants that use artificial intelligence algorithms to provide recommendation of the best quality embryos to transfer. Examples of this is Erica, the iDescord, and the Chloe. The microscope is one of the most important pieces of equipment in an IVF lab. Most of the microscopes in the IVF lab already come with a digital camera that can take photos and recording videos of the procedures being performed by the embryologists. These microscopes could be converted into smart microscope, but by adding processing capabilities to analyze the patterns of the images that they produce. An example of this is the AI-based sperm selection assistant SID, which can be used to evaluate all the motility patterns of each individual sperm in the visual field of the microscope in real time, and then select the best one to immobilize, guided by an overlay of computer-generated graphics into the microscope images. Also, it could be possible to use this kind of technologies to evaluate eggs before injection, and also to assist embryologists on different procedures such as oocyte retrieval and denudations. While making a smart microscope and incubators might help some of these challenges and improve results, the communication of these devices with each other and to a central processing unit is still missing. To solve this problem, IVF 2.0 have developed a software suite named DishS, which will allow the smart things on the IVF lab to communicate and share data, for example, pictures, videos, and outcome reports, with a central processing unit, which in turn will allow to achieve a highly reliable gamete and embryo traceability, generating detailed reports of the processes, of the embryologist's performance, and other KPIs, as well as collecting data to train future artificial intelligence IVF assistants. A smart lab will be equipped with IoT devices that allow the advance of monitoring the system and process that will track process of the complete procedure in real time. The pipettes and petri dishes will record the date and time of usage, along with the volumes of fluids used and their temperature. The laminar flow cabinets will keep track of the number of oocytes retrieved from the follicular fluid and after the denudation, as well as validating the name of the patient on the dishes, while giving alerts to the embryologists before any possible mistake happen. The laser ablation system will be capable of automatically determining the best place to perform the trophectoderm biopsy and will be shot automatically by an AI software. The smart IVF laboratory will be able to collect data from every instrument at any step of the IVF pipeline, including temperature, pH level, pressure, and other factors. All these collected data will be then used with big data, data science, and data analytic technologies to generate better protocols and treatments that will be custom tailored to be most effective for each individual patient. This will help clinics to improve their services and the medical doctors to make faster and better decisions that will allow the patients to get pregnant in a smaller number of attempts and at the same time cutting down on costs associated with the IVF treatment. Now, that's it. Thank you very much.
Thank you, Dr. Mendizaba. That's awesome. I'd love to see all of those products, and I hope to one day maybe be able to use a lot of those in our laboratories. My question for you is, have you done any studies yet with the combination of these IoTs or just any of the smart products within the lab to actually evaluate how much time is cut down on certain procedures? Because I would think if you could, like, stream them together as you showed, you could show that maybe, certain processes would be cut down in time. Maybe it would be better on the staffing levels, things of that nature. That's something we're always concerned with. Have you done anything like that, or know anybody who's looking at those types of things?
Regarding time, we haven't done that yet, but because the first step is always to get the information, gather the information, and then we will be able to do the analysis. That's why we are trying now the DishS platform as a beta in some clinics, so that will allow us to keep track of times and things like that. But we are right now doing a study where we relate the scores that we get from SID with the scores that we get from the oocytes, and we relate those scores with the embryo development. So we're about to release a paper with that information, and it is we believe that combining information from the different steps of the IVF pipeline will have a great benefit for all the patients ultimately.
I agree, and it's also going to just show us more KPIs and things that we probably never considered before.
Oh, yeah,
so thank
for
you.
sure.
Wonderful presentation.
Thank you.
Okay, with that, I'd like to go ahead and introduce our next speaker, Michael Skumial. Michael is a senior health care information technologist and operations management consultant. He's had this position for 20 years. He has 20 years of experience overseeing, implementing, developing, and innovating complex technology for major New York healthcare centers. Thank you, Michael.
Hello all, and thank you, Jean, for this introduction and the opportunity to talk about operations management in EIVF and specifically as it relates to embryology staff scheduling. So what is operations management? In any organization, including healthcare, at its core, operations management is the planning, the organizing, supervising of internal processes to keep an organization running smoothly. In IVF, these processes can be organized into communication, collaboration, staff scheduling, laboratory workflows and scheduling, operational and clinical scheduling, disaster management and business continuity, regulatory, organizing information and data, and content and document management. Specifically in EIVF, there's a great deal of resources designated to EMR applications, to devices, equipment, while often not so much to everyday processes and workflows that keep the organization running and that do not fit the typical framework of an EMR, for example. So IVF is especially challenging due to the unique and highly complex scientific procedures and complex clinical, laboratory, and administrative workflows that are more often than not plagued with information overload and
Hi, Michael. I'm sorry to interrupt, but we're having a little bit of trouble hearing you.
Yeah, is this better? I'm sorry.
In and out. Yeah, that's better. Thank you so much.
Thank you so much. I appreciate it. Okay, so organizations that pay attention and make an effort to improve these processes, as a result, greatly improve the efficiency as well as reduce the workflow and the work and stress associated with these processes. They also significantly eliminate errors and enable lab technologists to spend more time in the lab rather than performing manual administrative tasks, and thereby contribute to improving laboratory patient outcomes. So today we'll be, of course, talking again about staff scheduling. We'll be focusing on staff scheduling in EIVF and in the embryology labs. When it comes to staff scheduling, the process can be broken down into the following high-level workflows. So usually, if you think about how a schedule is created, it starts with, let's say, a month in the future, right? So when you are about to work on the schedule, by that time you would receive a number of requests from the staff. So submitting and processing schedule requests. The staff will send you requests for time off, for working days. Then at that point, next you would actually work on creating the schedule as a scheduler. Once the schedule is done, at that next you would distribute the schedule, and then your team members and the organization would access that schedule. Once the schedule is live, it goes to the next phase of maintenance, during which there are, of course, always last-minute scheduling changes, and the scheduler is responsible for making sure that these scheduling changes are captured and distributed again. So let's look at these workflows in a little more detail.
Michael, what you're saying is really important. I've seen your slides, but they are saying they still are having trouble hearing you. Is there any way you can adjust your mic or how close maybe you're sitting to it?
Thank you. I might be able to. Is this any better right now?
That sounds good. Yeah.
Okay. Thank you. My apologies for this. I'll try to make sure I'll speak a little louder. Okay. So the submitting and processing scheduling requests. So normally what happens is that the staff will either email the scheduler or they may communicate scheduling requests by word of mouth. And the scheduler's responsibility is to actually log all of these requests. And it also means that there's a certain queue, right, which matters, who was first, and this queue will be used to process these requests. So there's a pretty enormous amount of work that goes into just basically submitting and processing all of these requests and keeping track of who was first and in order to fairly assign it at some point later. So once the requests are all received, then the scheduler can actually begin working on the schedule. And the scheduler's responsibility is to ensure that there's adequate daily staffing levels, whether that means, what your total number of embryologists is per day that's needed. And that could be even broken down into the number of junior levels and senior level embryologists. Then we've also have business rules and requirements. There are, I'm sure there are a number of unique rules for each organization that the scheduler has to be responsible for meeting. And in addition, there's also obviously fairness and transparency. You want assignments and time off to be fairly and equally assigned for all of your staff. And that means perhaps keeping track of tallies for working days, holiday tallies, reports, and things like that. And then the requests, usually are broken down into three types of requests. You can have working day requests, which means if you do want to work and what you're responsible for that day. Non-working day requests, which are basically the days that the staff may not want to work on. And then time off requests, which are things like vacations. So that this, so we can, if we look at this process in more detail, we realize there are really a lot of steps, time-consuming steps that the scheduler is responsible for, and these folks are usually senior level embryologists, like embryology directors or managers, where I'm sure their time could be put to a much more better use than performing a lot of manual, repetitive steps. Just want to check in with you, make sure that you can hear me. Is that a little better?
Sounds great.
Okay, thank you. Okay, so once the schedule is completed by the scheduler, then the scheduler distributes the schedule, right? And the team members and the organization will want to access and view that schedule. And that means, there are many ways of that distribution, whether it's printing the paper and posting it in a lab, whether it's sending an email to everybody with the schedule, or maybe saving a document onto the shared drive. So of course, all of these methods are pretty commonly in use. And then again, team members and organization access that. So they basically go into a shared drive, go into the lab, or search for that latest email, and look at the schedule. And the schedule, will have, of course, information about who's working on each day, the assignment types. There may be abbreviations that are used. There's going to be color coding. There also will be holidays. And very often it will also include staffing levels and what the totals are for the day and any particular tallies, how many total embryologists you have, or if there are any senior embryologists assigned for that day. So that's what the distribution looks like. And what's interesting, what we find is that as soon as the schedule is published, of course, there are also immediately after that, there are changes. So once it's all published, right, we sort of go into this maintenance phase where there are last minute changes. Something comes up obviously in everyone's lives. You may have a doctor's appointment, or there may be, school event that you have to go to, and there'll be a great number of changes that will happen throughout the month. And these requests may come in as basically as a call to the scheduler, as an email, asking for a non-working day or for a day off or a vacation day. If the schedule is unable to accommodate, sometimes the embryologists are able to arrange a swap with another colleague. And that's, again, there's a number of steps that's involved in that, communicating with your colleague, of course, agreeing on what you're going to swap, and then, of course, communicating that to the scheduler. And those changes will also affect the staffing levels, right? Your staffing levels, your business rules. So the scheduler's job, again, is to kind of repeat that whole process and make sure that after all of these changes are made, the staffing levels are still adequate, and all the rules are met as required. And then, after that, all of it again, you have to still distribute an updated schedule. So depending on the number of changes that happen throughout the month, every single time there's a change, you have to send out the up-to-date schedule. So that's kind of what the scheduling workflows look like on a high level. And next, I wanted to talk about Fertility Connect, which is a operations management software suite for IVF developed by Health Connect Algorithms. This application suite has been in use by IVF centers for about 15 years. And throughout this time, the applications and workflows undergo a constant process of tweaking and further improvement to optimize the workflows. And I'll talk briefly about all of the different sections, and then again, we'll get into specifically into how embryology labs staff scheduling is done. So Fertility Connect helps with workflows and communication and staff scheduling and process automation. It provides an organizational portal, like an intranet for the staff. It helps with operational and clinical scheduling, laboratory information systems and laboratory workflows, disaster management and business continuity, regulatory management, content and document management. And it also has a mobile app component for staff schedules, scheduling requests, bulletin board for communication, contacts, and contact lists to your staff, and as well as disaster recovery and emergency alerts. So I have a screenshot here that's a backup, but will jump to a live demo, which we prepared for this presentation. And I will start with basically, again, kind of going back into the workflows and how you would actually perform these workflows in Fertility Connect. So as a embryologist, right, as an embryologist, you would, some of the things that you would normally do is, of course, submit requests. And then within this application, rather than emailing or talking to a scheduler or making a phone call, anything like that, you would actually submit your request right away into this interface. And you would basically just choose on a date and choose whether you want to work. If you want to have a time off, you can put a comment in there that only the scheduler will see if you have any specific circumstances surrounding your request. And then that point, as soon as you put this in, it'll actually populate that month that's open for submission. So when the scheduler comes in, all of these requests are logged, and therefore all of the manual steps in communicating those requests, keeping track of who was first, all of that is essentially eliminated and automated. So some of the other things that the staff member can view, it's besides obviously, of course, having an easy interface to view all of your requests and submitting all the different types of requests. You can there's a feature to automatically swap with your colleague if you can't get a time off day, and there are two types of transactions. There's a one-to-one transaction where you click on swap, and then you would choose the other embryologist and their day for swapping. That would generate an email and a message in the system, and then the other embryologist would essentially approve or not approve. If it's approved, the whole transaction is logged, and the schedule is updated. If it is not, the message just comes back that it was not approved, and there's a log that it was not approved, and there are no changes. There's also a three-way transaction where it requires managerial approval. So essentially the same process between the two embryologists, but the next step would be manager's approval to put a final approval for that swap to happen. There are also reporting options that provide individual embryologists with a detailed report of all of your requests, all of your working days, all of your non-working days, holidays. There are assignment tallies if you want to see your personal tallies as far as how many assignments you work on the different types of the week, or how many assignments you have worked for any period of time. There are also working day tallies and holiday tallies. And the holiday tallies will essentially will let you know how many holidays you have worked. So let's say if you want to see how many Thanksgiving days have I worked in the past 5 years, you'll be able to kind of get all this information. And as an embryologist, you only see your personal tallies, by the way. The scheduler can view everyone's tallies, but each individual embryologist can only view their personal tallies. There's all of the requests are basically abbreviated. There's a map of that you can mouse over, get like a full definition. There are up to 3 assignment types. There can be more, but usually we found that up to 3 are enough. Holidays and weekends are all color-coded. There's a time off map. There's also a scheduling expectation, which is like the business rules, and the comments are all color-coded. So as embryologists, you have access to all of these features, and you can also easily and quickly access your schedule and also the entire team schedule online, rather, of course, than obviously going to paper or retrieving a document on a shared drive or searching an email for the latest version of the schedule. So next, I will talk about what the workflow looks like for the scheduler. So we'll click on embryology staff scheduling. And when it comes to the scheduler, usually what would happen when you talk about creating the schedule. So let's say for example, let's say where I am the scheduler, and the next month I actually have to create is the February 2023. So when you start, actually, what happens is you change the status to in progress. So you prevent anyone from making changes. Obviously, as you are about to work on it, you'll change it to in progress, and that kind of locks the schedule. And then the nice thing is that you will have all of your requests already in, so you don't have to go through your emails, through your notes, talk to anybody. All of those requests are already in, and you will see them here. Some of them that will have any comment, you can just mouse over, and you will see what the particular comment may be. So it's kind of it's nice to have that and not have to re-enter that information. So your job as a scheduler, again, is to make sure that you have adequate staffing levels based on the business rules and the requirements of the lab, and also that you actually fairly and transparently assign days off and working days for your team. So one of the options that you have is, of course, you can manually assign all of these. As you make the changes, you will see here at the bottom that these alerts will start disappearing. So you can program the staffing levels per day, depending on your lab. I also do want to mention that I had a chance to connect with Dr. Alikani and an opportunity to talk about her work about figuring out what these staffing levels should be. So there's a great amount of work out there that, of course, Dr. Alikani contributed to, and then there's other resources as well about what these staffing levels should be per day for each lab, based on obviously certain variables. And the interesting thing is, after you figure those out for your lab, you can actually enter them into this application, and it will keep track for every single day whether you actually are meeting these requirements or not. And what you see here at the bottom is basically for this month I am about to begin working on. I see here that all of these reds means that I am not meeting these requirements, and these especially need attention. And ideally, when I'm done with the schedule, all of these reds should disappear. So that's very helpful. There are a number of alerts, business logic, and unique rules that can be entered into the system that will help you with making sure that you have adequate staffing levels, and also to eliminate any scheduling errors, like assigning somebody who's off and things like that, and a whole bunch of different types of errors. So let's say I'll give you one example. So let's say if usually holidays are a challenge, right? So I do want to mention, by the way, that these names are all obviously made up, and there are names of former first ladies of the United States. So in this example, let's say if I'm looking at a holiday, which is highlighted in red, I see here that we have a good number of requests, right? And again, the challenge is I don't have here enough of embryologists assigned to this day. The challenge is that a lot of people, again, do want to have this day off. And how do I, as an embryologist, fairly assign as far as who should get it or not? So one of the tools that what you could do, you click on the holiday tallies, right? You click on the holiday tallies. You can select the entire team or just one embryologist. In this case, I'll want to see everyone's tallies, and then you can choose any specific range. If you want to go back to the past 10 years or the past 2 years, whatever they may be, I'll just go back to 2019, and we'll go into the end of the current year. And then what will happen is that you can actually see the holidays and the tallies for each one of your members. So in this case, it was Lincoln's Day, which was a holiday celebrated in many of the states in the United States. Not every single state, but many of the states actually have this as an official holiday. And I can see here by kind of quickly looking at, and I would probably look for the person that has actually worked the least on this day. And I see here, for example, most people have worked at least once, except for two, and one of them is Adams, and the other one is Lincoln, right? So that's probably those are the folks that I would want to assign to work actually, and not get this holiday. I know they've submitted a request not to work on this date. So now that I actually know quickly who again should be working or not, I can try, for example, Miss Lincoln. She would put in a request to not to work, but I see she actually hasn't worked a single day. So maybe she's a good choice. And now actually, when I look at it, she's also put in a note for special circumstances. So, there was another person that basically also had a 0. So in this case, looks like, again, the best choice would be Miss Adams. And then in that case, I would just click on that and change that to a working day and submit it. And then what you see here is that red disappeared. So, at this point, we're happy. We have we haven't adequately assigned the embryologists to make sure that we have, again, enough of embryologists, and also fairly assigned the working day on a holiday. So, that kind of gives you an example of how that works. So additionally, you have an easy way to kind of browse back and forth for the different months. Once you complete the schedule, you would just essentially click on change the status to publish date, and that actually publishes the schedule. At that point, it becomes available to the entire organization to view what the schedules look like. And you sort of go again into this maintenance mode. I do also want to mention that, you have, in addition to sort of manually assigning these, which is a lot. If you think about it, if you have 20 embryologists in a month, that's about, 30 days, and that's about 600 slots that you have to actually fill out every single time, which again, it's work done by senior embryologists, and their time is very valuable. So one of the options in this application is to auto-generate the schedule. And the way it accomplishes that is that you can preload a template right, for each employee, and that it will follow that template. It'll follow all of the business logic. It will also use a random generator. It can also utilize historical data to fairly assign it, and also there's AI module to also sort of fairly and randomly assign the and automatically create your entire schedule for the month. So with just one click, you can click on that and fill out the entire schedule, and then still be able as a scheduler to kind of look at it and make your kind of final tweaks. You'll also have an option to run a detailed report. So anything that happens within the system is there's a trail, audit trail that you can view, and it's timestamped, and every single action is recorded by every single user. So whether it was canceled, whether it was assigned, change in queue, or swap, you can go back and see every single thing that happens for that particular assignment. There's no question as far as going back and basically saying, oh, well, something happened, I'm not sure what happened. You'll be able to kind of retrace the steps. There are assignment tallies, so you can see, again, for different type of assignments, what your staff, what the numbers are, what the tallies are over a certain period of time, and see how many, let's say, how many working days, how many times were you responsible for anything. There are also day of week tallies, so you can, if you want to know how many Sundays this person worked, or how many Mondays, or how many weekends in total, again, to fairly assign the assignments for your staff. And there are also holiday tallies, of course, we talked about those. So as in terms of assigning, again, we have three types of assignments. So we have one assignment, we also have a second assignment, and the third. And that could mean, it could basically, you have the ability as a scheduler to create very simple assignment as working and not working, or you could get more detailed and say working and then responsible, for example, for quality control for that day, or be responsible for answering the alarm if the alarm happens. So, you can get as simple or as obviously as detailed as you'd like to.
Michael, thank you so much. All of this is fantastic for all of our labs. I know that we have so many people on here, and they're probably in lab staff from, 3 embryologists up to 30 embryologists. So I know that for all of them, they can take away from this how difficult, one, it is to staff a lab when you have so much fluctuation like we do in the IVF world, and so much different complexity. So to have a platform like this where we can review historics, fairly staff our labs so that we're not overburdening one person to another as far as holiday time. So thank you so much. I know that we have a video that is supposed to show later on in this. Dr. Alison Campbell and Ineabelle Collazo have put together a really great one about their real life experiences.
So I'm Ineabelle Collazo. I'm the embryology laboratory director for the IVFMD labs in Florida.
Hello, I'm Alison Campbell. I'm the Chief Scientific Officer at Care Fertility. It's a network of clinics in the UK. We also have a clinic in Dublin, Ireland. And so talking about QC, well, it's really important that clinics, IVF laboratories have dedicated QC and assurance. And I think it's changed so much over the last few years. It used to be very much forms and checklists and folders of information, and we'd go around the lab with a pen and paper, filling in the boxes and maybe getting to analysing it at the end of the month. Whereas now it's much more cloud-based, it's a lot of data, it's much better, so much more information to collect and to retrieve very rapidly. So we now have these tools at our fingertips so that we can really analyse what's happening within our critical equipment, within our laboratories.
IVFMD currently has 4 IVF labs in the South Florida area. They're quite apart, and we have 6 andrology labs.
So because we work in a group of clinics, a network, having a cloud-based system for quality control, it's really important and it's really useful because we can look remotely to see how the clinics are performing in terms of the the critical parameters on their equipment and all the QC elements that are measured, we can compare and contrast between the clinics. The laboratory managers can access data from their own clinic very easily. So they can look out for trends, they can do their analyses from home. And so it's a much more convenient way of working.
One of the most important things in the IVF lab is the quality control. We start our morning by QCing labs, all the laboratories, first thing before we start doing our work, checking the gases that goes to the incubators, the temperatures in the incubators, making sure that everything is running as it's supposed to be before we even attempt to open an incubator or start doing a procedure. Everything has to fall within the ranges that were specified. I can see all the QC from all of my 6 locations for andrology and for IVF labs from my desktop or even my mobile app, which is critical for me in order for me to manage all the laboratories.
Well, having all this data related to quality control is really important for the users, but it's also really reassuring for the inspectors. We have regular inspections in the UK from the Human Fertilisation and Embryology Authority. So having this so readily accessible is quite impressive and reassuring for the regulator. And we also do inter-lab inspections because we work as a network. One of the laboratory managers from one of our clinics will go to another one, and we have templates to do an inspection just to make sure everything's running smoothly. And again, these QC charts and the cloud-based system really useful for that, so that we can inspect each other and make sure everything's top of its game.
Yeah, I look at everything remotely. I'm stationed in one location, which is our Miami location. So it's easier for me to go to the lab and look at everything. But for the locations that are, for example, 125 miles away, I just go through my phone and I just select the location, and I'm able to see everything that is going there. Also having the mobility and the ability of having a mobile option, I can set up for when parameters are out of range. I use my QC for the andrology team, for example, their RQ beats, their motility clips, all that stuff are connected over there. So the moment that something is out of range, I get an immediate alert. It's addressed instantly. So we have a protocol in place for that, where andrologists cannot start testing unless they are within range. So it is important, it's critical, and it's nice to be able to see everything from one place.
So we have an obligation to monitor critical equipment continuously. And so this data is really vital. But on top of that, we like to do independent assessments because we can't be wholly reliant on a continuous automated monitoring system. So we do these human checks as well, which we can also record on the QC cloud-based system.
So currently, in order for me to be able to see our QC from each location, we do have a 24-hour monitoring system that alerts us at any time if something is not functioning correctly. We have PharmaWatch, which is our monitoring system. We also have CryoSentinel that for our tanks and stuff like that. The beauty of it that right now we are capturing the data in the morning. We are manually entering into an app that is called Reflections. So we're doing that every single day for different type of equipments, and it varies per day what we're monitoring. The beauty of our system that we have right now is that PharmaWatch will be integrating with Reflections, which means that PharmaWatch will be communicating with them so that we don't have to enter that data manually.
And so this adds an extra layer, I think, of reassurance. You know, and an example might be we are continuously monitoring the temperatures and the levels of liquid nitrogen in our dewars, in our cryopreservation room. But also we will add on physical and manual checks just to make sure that the system is working correctly and that we're not missing any early warning signs of condensation on the tanks, for example, or pools of water on the floor of the laboratory.
The beauty of the two systems is now PharmaWatch will be integrating with Reflections, and that will allow us to not have to enter the data ourselves manually. So PharmaWatch will send that information to Reflections, avoiding the staff having to enter that, which, you know, is kind of a time saver for them, and giving us at the same time live data. So it is important tools for us to have in a day-to-day work day in a laboratory, and also when you have multiple locations, as we do, that is critical. So it's been useful. In Florida, not too long ago, we had a hurricane hitting our West Coast. We have, all 4 labs that we have for IVF had the potential of being affected, but primarily the one is in the West Coast of Florida, in the Naples area, for those that you're familiarized with the US. So that one was critical to have all our alarm systems, let it be CryoSentinel, monitoring our tanks, let it be PharmaWatch, monitoring every single equipment, and being able to see everything through the whole storm passing, being able to understand that if we got a flicker in power, it will detect it, it will alert me right away. That was a huge relief.
So I think together we have the cloud-based system which collects all this information, but we also feed into that data from all different places, so we can get the very best from the system and make sure that we are absolutely as safe as we can be for our patients.
Thanks again for all of you attending. We have a lot of questions for our panelists. So if I can get all the panelists to come back to their cameras and turn on your mics, and we'll get started with a little bit of Q&A. Okay, so I'll go ahead and ask the first question here. It's back to Dr. Alikani. And the question comes from Francisca Barr from Denmark. She's a global manager for Vitrolife. How often do you recommend pH measurements? Is continuous the best way?
Yeah, I mean, this is a question that has been debated over and over. I would say that my own preference would be not to measure pH continuously. I don't think there's a need for that. I think that measuring pH once a week, or when new media are about to be put into place, or when there is drift in the CO2 levels and you want to confirm the CO2 levels with a pH reading, that would be the time to run pH. If you want to keep your sanity, that is.
All right. Cassie,
I did want to, Jean, I know you're looking at some questions. While you're looking for the next question to pose, I did want to let everybody know that Dr. Campbell and Inya's video will be available on the website. So please visit, please watch it. It's going to be a great video to watch, very informative. I'm just so sorry that we ran out of time today.
Okay. The next question is actually for Rusty Pool. It comes from Francisco Barr. Does AAB have specifications for reporting all this data?
AAB?
Yes.
No. No.
All right. Very short and sweet.
Yeah. No.
All right.
All right. Let's see. Who is this for? Alikani, Dr. Alikani, another question for you from Melissa Zorn. How does an inspector look through QC during an inspection from a platform like Reflections?
Yeah, very good question. We've done it in different ways with different inspectors. There are inspectors who are very computer savvy and interested in just going right into the app and with someone sitting next to them from the lab and navigating through the app. And that has worked beautifully because they can essentially see whatever they want. They can have us graph things. They can have us show them logs from any time, anywhere, on any instrument. So they like that. There are others who will say, could you print all of your reports for me for, the month of November and, August. And then we've done that as well, where we show them the actual printed reports. I've dealt now with the Department of Health in New York State. I've dealt with the Joint Commission and the CAP, and all of them have accepted this as a way of recording, documenting quality control.
Thank you. That's great. And then I'm going to pivot to Dr. Rusty Poole. There's another question for you. It doesn't say who it's from, but the question was, did you say 80% of ransomware threat agents do not return data even if you pay the ransom?
That's correct. 75 to 80% are not going to, what they don't want to do is generate one more trail for you to catch them. So there's no point in them communicating with you after they made the transfer. So they're not good people.
And generally, is that data still shared? Have you bought yourself any time or any security, or is it just, you hope that 20% gets returned to you and it's safe and secure and never used again? That's
correct. I mean, the best way to do it is avoid them hacking you. There's really, once you get hacked and it's a ransom deal, it's a tough proposition. But yeah, they're bad boys.
Okay, well then I'm going to piggyback on that and ask this next question to Dr. Gerardo Mendizaba. The question is, what about the security of IoTs? Has there been any issues with that?
Yeah, that's something that's a really big concern with this IoT technology, because as I say, the things have to communicate with each other and to central unit processing, and they have to use the regular channels. So if they're connected wireless, of course, there's a risk that somebody could, get that data from the air with, some kind of sniffer, things like that. But the good thing is that now we have very good encryption algorithms and methods. So that risk will be alleviated if the people do the things right and put the extra layer of security. That should not be a concern at all.
All right. Let's see, where do we want to go next? I like to add the reflection questions. So we talked about reflections being accepted by inspectors, and you said most, Dr. Alikani, that most inspectors will go in and sit down with the computers and everything else. Is reflections limited to just equipment checklists?
Well, equipment and checklists, you can be very creative or
right.
very creative and use it in any way that you think the lab would benefit. I think that flexibility is built in, which is a nice, really nice feature of this application. Nothing is dictated, so you are free to set it up the way you want it. And so I can imagine, and there are people who use it, and there are in other industries as well, who use it in unconventional ways. Checklists are particularly interesting because you could give them a twist and look at all kinds of things. So it's really quite flexible.
I know I don't know about Cassie, but when we set up our dashboards, they're pretty individual. There's some standard things, obviously, everybody looks at as embryologists or supervisors, but then there's stuff like, I'm like, if you want everybody to walk around the lab and turn off the microscopes, then let's put that on the checklist. Let's, put that on there. And it, obviously it will complete itself and be green at 100% once everybody's done everything. So if they need a reminder to do that, then let's put that as a task. So you're right, pretty much anything can go on there as a checklist item, not just equipment, QC, as we think about temperatures and things of that nature.
Yes, absolutely right.
I agree, Jean. Yes, we use it as kind of a reminder list too. Did I turn off the lights? Did we set the alarms? Did we mop the floors? It's
Right.
just good. We humans need the checklist to say it's done. Yes.
I agree.
I have one last question here for Michael. We have some panelists that want to know, does it work for other departments in the IVF lab? Meaning, if it's a big clinical setting, can we also include clinical personnel in this staffing model?
Cassie, can you hear me?
Yep.
Great. Yeah, certainly. Yes. So Fertility Connect has modules for all the different types of teams within IVF. And that includes nursing, medical assistants, physicians, of course, the laboratory teams, endocrinology, whether it's embryology or andrology, as well as administrative teams within IVF. And of course, a lot of the challenges, scheduling challenges, apply to all of those different teams. There are obviously unique business rules and requirements for each, and the systems offers enough flexibility to be able to specify these rules uniquely for each team. And so again, if you think about like on a bigger scale of your organization, if you are able to improve this process for one team, not just one team, but for all of your teams, you really can eliminate a lot of the manual, tedious work and make everybody happy.
Great, thank you.
Thank you. So this is going to wrap up, but there's one more question for Rusty Pool, and the question is, how much does it cost to run or open a goat farm?
Depends on how many goats, but a bunch. The land alone will kill you.
Oh my gosh, thank you. Thank you, everybody. Thank you for our speakers. Thank you for everybody joining. Thank you for joining this special seminar today, again, sponsored by Reflections app, and that comes from the IVF QC software platform. We hope you enjoyed this session. Look out soon for our podcast to accompany this session. And in fact, you can listen to all of them and rate our podcast as well. I3's next session is about the next generation of embryologists, the Gen Z. Who are they? What do they want? And how can we work to our best ability with them? Much has been said about this young generation of clinical embryologists. So let's meet a group of highly dedicated and talented young professionals at the start of their already impressive careers. They'll be joined by the bromance of Dr. Campbell Pomeroy and Huey Hune, and a human resources specialist, Angela Regan, who knows all about digital natives called Gen Z. Thank you again for attending today. And continue to join us for I3 sessions. Take care.
Thank you.
Thanks, everyone.
Bye.
Thank you.
Bye.
Thank you. Bye-bye.